Okay, other movements are also there. For example, whenever you are swallowing something, that is also a movement. Whenever the digestion is taking place, that is also a movement. Okay, like these different types of movements are there, which are together done by the muscle fibers. Now, what are the characteristics of the muscle fibers? So you can see irritability is there, contractility is there, extensibility and elasticity. That means whenever you are giving stimulation to a muscle fiber, it will respond to that stimulation. That is called as the irritability. Then contractility means enough stimulation, if it is there, then it will contract. That is the contractility. Extensibility means it can be stretched beyond its length. Especially it is seen in case of smooth muscles. Next, elasticity, ability to return to the resting stage once the stretch or the force is removed. Okay, so these are some important characteristics of the muscle fibers which are there in our body. Now, if you take a individual muscle fiber, now the muscles are made up of muscle fibers. Any muscle if you take, they are made up of muscle fibers. Take one individual muscle fiber, it is nothing but a cell, and that is called as myocyte. Okay, जो muscle होता है, how they are built, they are made up of muscle fibers. If you take one, now they appear to run transversely across the whole muscle fiber. Okay, and because of this only the particular arrangement, the striations can be seen. Next, between the two Z bands, it is called as sarcomere, already I have mentioned. Okay. Next, the mitochondria are numerous and substantial amount of glycogen are present. Okay. Now, glycogen provides energy for the contraction of the muscle. So, what is this? Can anyone tell me what is this? ये जो पूरा structure दिखाया है, what is this? How many muscle fiber? One individual muscle fiber. ठीक है. Inside that, ये सब क्या है? What are these? Myofibrils. ठीक है? So I hope you have understood. Now you have taken out one myofibril. ठीक है? बहुत सारा myofibril से you have taken out one myofibril. Then you can see this alternate dark band, light band, sarcomere, all those things can be seen. ठीक है? Next, inside this myofibrils, can you see this small cut section? These are nothing but the myofilaments. ठीक है? So till now whatever we have discussed, that is up to myofibril. I have not shown you the myofilament. Now we will see the myofilament. Okay, as you can see, these are the bands, whatever I have mentioned just now. Same thing. Okay, now for that myofilament what you need? Ultrastructure. Ultrastructure means electron microscope structure. ठीक है? तो myofibrils are the structural and functional unit of muscle fiber. ठीक है? Next, myofibrils are arranged longitudinally and runs throughout the length of the muscle fiber, which I have mentioned earlier. Myofibrils consist of bundles of myofilaments. जो myofibrils होता है, that means one myofibril, inside that bundles of myofilaments are present. ठीक है? Now, how the myofilaments are made up? Now, this myofilament consists of thick myosin 2 protein filament and thin actin protein. Okay, that means thick and thin filaments are there. Thick filament is myosin filament, thin filament are also called as actin filament. Okay, तो इसका जो arrangement है alternately होता है. Okay, thick, thin ऐसे करके alternate arrangement होता है. That's why you can see the striations in the muscle fiber. Okay, now arrangement of these thick thin filaments gives striated appearance to the muscle fiber as observed under H&E. But in H&E you can't see the these myofilaments. Okay, for that, as I said, ultrastructure is required. Now, structure of myofibril. So already I told you in the structure of myofibril, actin and myosin filaments are there. Let me show you the diagram. Yes, this is the, so this is the thick filament. These are called as myosin. This is the thin filament, these are called as actin. So see, alternate arrangements are there. Thick, thin, again thick, again thin, again thick. Okay, like this it is there. That's why the striation appears. Okay. So that is all about the skeletal muscle fibers. So we have gone into the smallest part of the skeletal muscle. इससे small कुछ नहीं है. और ये जो individual fibers, actin myosin filaments है, these are nothing but the different types of protein. If you go into more detail, then you have to study biochemistry. उसमें amino acids आएगा. Okay, most commonly जो है this one, this is troponin. okay, which is present over here. So further if you go, then you have to discuss about the biochemistry. Okay, so that is all about the skeletal muscle fibers. Next, in skeletal muscle fibers, you know that three different types of fibers are there based on the contraction, based on the speed of the contraction. These are called as type 1 slow fibers, type 2 fast fibers. In between them, another type of fibers are there, and these are called as intermediate fibers. Have you heard about it? Whenever I am talking about skeletal muscle, okay, so you know where the skeletal muscles are present in our body. Okay. Now, whenever you are using the skeletal muscle, there are some skeletal muscle which you can use for longer period of time, and that time there will be no fatigue. For example, right now you are sitting, or suppose tomorrow is Sunday. Suppose you are going for watching a movie. Okay, at that time movie ka timing suppose 2 hours 3 hours hai. So you are sitting, you are relaxed, you are watching the movie, but at that time your postural muscle or the muscles of the back, they are acting, they are working, but you are not getting fatigue, right? So that is one type. Another type, suppose you are free, okay, you don't want to study, you are playing games continuously in your phone, PUBG or some other games, you know better than me. So kya hoga? Can you play for 3 hours 4 hours? No, within half an hour you will feel pain in your hand. वो भी मसल है, ये भी मसल है. अभी समझ में आ रहा है difference? And some in between them. No, this is the fact. In between them, there are some muscle fibers. Okay. For example, you are going for a long walk. So you can walk for, I think, for 20-25 minutes. After that, or 30 minutes, then you will get fatigue. So that is in between. Okay. So all are skeletal muscles only. But the way they are contracting, okay, the force of contraction which they are providing, in between them, the differences are there. Have you understood? So first is the type 1 or slow fibers. These are also called as red fibers. तो उसमें क्या होता है? It contain large amount of myoglobin. ठीक है? In this particular type of fibers, large amount of myoglobin are there in the mitochondria. Now they are also called as slow twitch fibers. Why? Because they contract very slowly. And that's why they are not prone to fatigue. Fatigue नहीं होता है in those type of fibers. Okay, they generate less force of contraction but produce prolonged contraction. Example, postural muscles of the back. Okay. Next, type 2 fibers are also called as fast fibers. So they are also called as white fibers. They are thick fibers and they contain very less amount of myoglobin in the mitochondria. But the amount of glycogen are more in those type of fibers. Okay. So these fibers are high twitch fibers and can generate strong force of contraction. Okay, so the force of contraction are strong and it is required for doing that particular task. Okay, and they are very prone to fatigue. Okay, so what will be the example? Already I have given you the example. The example will be the muscles of the hand. Okay, or extraocular muscles of the eyeball. Okay, extraocular muscles of the eyeball, muscles of the hand. In our eye also, see whatever movements you are doing without turning your head, without moving your head, you can see above, you can see down on both the side. Try it. You can do it. कैसे होता है? You can move your eyeball. That is done by the extraocular muscles which are attached to the eyeball. 7 extraocular muscles are there. ठीक है? So in head and neck portion, we will study about that extraocular muscles. Very important. Okay, next is the intermediate. Okay, so between the comparison, if you see the comparison, if you draw the comparison between the red fibers and white fibers, this is the points. Okay, these are the points, or you can say these are the differences between the red fibers and white fibers. So I'll request everyone to go through it so that you can understand about the difference between the type 1, type 2 or fast and slow twitch fibers. Next, what is this? This is the connective tissue covering of skeletal muscle. Okay, connective tissue covering. So as I said, the connective tissue is covering the entire skeletal muscle. Now, it not just covering the entire skeletal muscle, but it is also covering the bundles as well as it is also covering the individual muscle fiber. Okay, and the names of those coverings are different. Now, if I ask you, what is the importance of this connective tissue covering? Anyone in the class, if you are using your mobile phone or tab, anything, please keep it inside the bag. Anyone? Okay, don't use tab or mobile phone inside the classroom. Not allowed. I will not repeat. Whenever you are free, you can use. Inside the classroom, practical hall, you are not allowed to use all those things. Okay? Now, why these connective tissue coverings are there?
To hold a bundle.
That means support. Yes, to provide the support. Now, but what is the main function? Yeah, protection is there, support is there. Along with that, the other main function which you have not mentioned is, along with the connective tissue covering, the blood vessels, nerves are coming into the muscle fiber. Can you see in the diagram blood vessels, nerve fiber? Ye dekho. Can you see? Okay, so these are also required. Blood vessels, nerves, these are required for the muscle fiber. So they are bringing the connective tissue covering, they are bringing the nerve fibers, muscle, blood vessel to the muscle fiber. Okay, now, so this is entire muscle, as you can see. Okay, this is entire muscle. Now this entire muscle is covered by a connective tissue covering, and this is called as epimysium. This is called as epimysium. Now, if you take the skeletal muscle, as I mentioned earlier, it is made up of bundles of muscle fibers. Okay, so many bundles of muscle fiber. Okay, you can see so many bundles of muscle fibers are present. Now, they are covered by another connective tissue layer, and that is called as perimysium. Okay. Next, if you take individual muscle fiber, see this individual muscle fiber, they are also covered by a connective tissue layer, and that is called as endomysium. Okay, so like this entirely it is covered by connective tissue covering, which is required, okay, to provide support, then protection, and along with that it will carry the blood supply, nerve supply to the muscle fiber. Okay, so here you can see the individual muscle fiber surrounded by delicate connective tissue, that is called endomysium. Individual fasciculi, okay, the fascicular arrangement of the muscle fiber, that individual fasciculi, that means so many muscle fibers will together form a bundle, that is called as fasciculi. So that fasciculi is covered by perimysium, and entire muscle is covered by epimysium. So that is all about the skeletal muscle. Next is the cardiac muscle. Now in cardiac muscle, what you have to know? In cardiac muscle, first of all, the muscle fibers which are there, they will branch with each other. Then striations are seen, but it is less prominent. Striations dikhai deta hai, but they are less prominent. Next, the nucleus which is there, it is centrally placed, single nucleus. Okay. In between the individual muscle fibers, the connection is there. A special type of connection is there, and that is called as intercalated disc. One muscle fiber, another muscle fiber, they are connected to each other by a special type of connection, and that is called as intercalated disc. That is the unique identifying feature for cardiac muscle. Okay. Now here identification is very easy. Okay, but you have to understand the structure of the intercalated disc. Now in intercalated disc, two components are there. As you can see, intercalated disc are the complex adhering structure. What it will do, it will connect the single cardiac myocytes to an electrochemical syncytium. Now these discs are responsible mainly for force transmission during the muscle contraction. So transmission of force, that means from one muscle fiber, force is transmitted to another muscle fiber. Okay, with the help of this particular intercalated disc, so that they can contract in a syncytium. That means they can contract together. That is the unique property of cardiac muscle fibers. Now, as I said, it is having two components, okay, and three parts are there. Fascia adherens, macula adherent, and gap junction. This macula adherent is also called as desmosome. In physiology, I know you have studied about the cellular junctions. In physiology, I know you have studied about the cellular junction. Okay? So, fascia adherens, desmosome, and gap junctions. These are the components which are present in the intercalated disc. Okay? Next, these are the way how the intercalated discs are formed, and you can see two components are there, transverse component and longitudinal or lateral component. ठीक है. Now in case of transverse component, it is perpendicular to the myofibril. So if you compare the myofibril, okay, that means a particular myofibril, then this transverse component will be perpendicular, and the way they are arranged, it appears as a staircase appearance. It will appear as a staircase appearance. Okay. Next, the lateral or the longitudinal component, they will lie parallel to the muscle fiber. Okay. Now this transverse component consists of fascia adherent and desmosome, while lateral component present only the gap junction. Now this particular gap junction, this particular intercalated disc, the allow action potential to spread between the cardiac cell, okay, permitting the passage of ions between the cell. This is the requirement. That's why the intercalated discs are present. Because of the presence of intercalated disc, what will happen? The action potential can spread from one particular cell to another. So here you can see fascia adherence, desmosome and gap junction. These are the three parts of a particular intercalated disc. So fascia adherence में क्या रहता है and what is the function? Now it gives attachment to the thin filaments of the terminal sarcomere. Okay, so this is the main function of the fascia adherent. Now in case of fascia adherent, it is similar to zonula adherents of the epithelium cell, which you have studied earlier. It binds adjacent myocyte with each other to form the cardiac muscle fiber. Okay, so adjacent myocyte, that means one muscle fiber, another muscle fiber, जो attach करके रखता है, that is done by this particular component, that is the fascia adherent. Okay, and इसका function क्या है? It mainly gives attachment to the thin filaments at the terminal sarcomere. Next, desmosomes are there, are also called as macula adherents. Now इसका function क्या है? They will bind the adjacent myocyte with each other, okay, and they are present in both transverse and the lateral component of the intercalated disc. Lastly, the gap junctions, they are present in lateral component of the intercalated disc. Okay, they provide ionic continuity. That means the ions from one cell or one cardiac myocyte can go to the other with the help of these gap junctions. Okay, that will help to spread the action potential rapidly. Though ions are moving, so the action potential can spread rapidly from one cell to another with the help of this gap junction. Now here you can see the diagram. So this is the cardiac myocyte, or you can say cardiac muscle fiber. Okay, just see how is the appearance. So you can see the striations, you can see the nucleus, centrally placed nucleus. Then you can see these are the intercalated disc. Okay, these are the intercalated disc. Now if you see a electron microscope structure of the intercalated disc, up to the intercalated disc can be seen. Beyond that, the structure cannot be seen. For that you need electron microscopy. Okay, so under electron microscope, if you see the structure, this is the appearance. Okay, so here you can see the transverse component and you can see the longitudinal component. ये देखो, this is the transverse component, this is the longitudinal component. Okay, so here you can see the fascia adherence, you can see the desmosome, and you can see the gap junction also. Okay, so this is an easy diagram which I have selected, and it will help you to understand about the structure of the intercalated disc. Okay, next the myofibrils, which are there in that striations are present, same actin, myosin, filaments, everything is there, but the appearance, that means the prominency are less as compared to the skeletal muscle. Means it is less prominent, but everything is same like the skeletal muscle. Okay, next T-tubules are also there. Have you heard about T-tubules, anyone?
Yes.
क्या होता है T-tubules? I know you have heard, you have studied.
Cardiac muscle, there are some tubules that present longitudinally.
What is the function?
They are receptor present and stimulus the receptor in tubules for calcium.
Not exactly. See, T-tubules are present in case of skeletal muscle also. ऐसा नहीं है कि सिर्फ cardiac muscle में है, skeletal muscle में भी T-tubules रहता है. But the difference is the T-tubules which are present in case of cardiac muscle and skeletal muscle, okay, they are forming a particular structure. In case of skeletal muscle, it is called as triad. In case of cardiac muscle, it is called as dyad. Okay. So in case of cardiac muscle, each T-tubule in the cardiac myocyte is surrounded by a small cistern. Now what is this cistern? It is a dilatation of the sarcoplasmic reticulum, and it is present only at the level of the Z line. Okay, so one T-tubule and one sarcoplasmic reticulum. Okay, so two structures are there, that's why it is called as dyad. In case of skeletal muscle, okay, two cisterns are there on either side and one T-tubule is there, that's why it is called as triad. Okay. Now these T-tubules, what are these? Those who have not studied about the T-tubules, okay, please see what is T-tubule. These are nothing but the extension of the sarcolemma. They will penetrate the cardiac muscle where at the level of the Z line or at the level of the sarcomere, in between one sarcomere. Now these cardiac T-tubules are highly branched invagination of the cardiac myocyte, especially in the sarcolemma, that are rich in ion channels import for excitation, contraction, maintenance of resting membrane potential, then irritation, regulation, signaling, transduction. So these are some important functions which are done with the help of the T-tubules. Okay, so mainly क्या करता है जो T-tubules है, जो impulse है, is carried deep into the cell or into the muscle fiber with the help of the T-tubules. That's why the T-tubules are present. T-tubules means transverse tubules. Here you can see, so this is the sarcolemma, okay, and see these are the T-tubules. This is one T-tubule, okay. So these are the invagination from the sarcolemma, and it is in case of cardiac muscle, the T-tubule, one T-tubule is present in one sarcomere, okay. So one T-tubule and this cistern, that is the dilatation of that particular part, will form a structure, and that is called as dyad, okay. Same in case of skeletal muscle, दो cistern रहता है and one T-tubule, that's why it is called as triad. Okay, next this is the diagram which you have to draw in your practical notebook. Okay, this is the diagram which you have to draw in your practical notebook. This is the diagram showing the cardiac muscle fiber. Okay, now here you can see these are the fibers. Now you can see these are branching with each other. Then you can see the centrally placed nucleus. Then striations are there, but they are less prominent. Okay, and then you can see the intercalated disc. ये देखो intercalated disc. Okay, along with that you can see a perinuclear halo is also present. This under H&E staining around the nucleus, a bright zone is there where most of the cell organelles are present. Okay, and that is called as perinuclear halo in case of cardiac muscle. Okay, so previous diagram which I have shown you, that is the schematic diagram which you have to draw, and these are the microscopic structures, okay, in high different magnification. तो ये 10x में है, ये 40x में है, and this is in under 100x. Okay, last is the smooth muscle. Okay, so smooth muscle, why it is called smooth muscle, already you know that, because striations are not there. Okay. Now these are also involuntary muscle because they are under control of sympathetic and parasympathetic nervous system. What is the location? Smooth muscle का location क्या होगा? Here you can see gastrointestinal tract, blood vessel, respiratory tract, urinary tract. Okay. Now please note that here I have mentioned about the respiratory tract. Can you see? Today we will study about the trachea. Trachea is a part of. Trachea is not a part of smooth muscle. It is a respiratory tract. ठीक है, तो उसमें स्मूथ मसल आएगा. Okay, a part is there where smooth muscle cells are present in the trachea. So you have to follow this, because after that when we will start the trachea, this will help you to understand the smooth muscle fibers. What is the appearance in the trachea? Next structure, spindle shaped. Okay, size it varies depending on the location wherever it is situated. So in case of blood vessel, you can see 15 micrometer, and in case of uterus, whenever the pregnancy is there, at that time it becomes 500 micrometer. So the length varies in case of smooth muscle fibers. They are not same everywhere. Next, as I said, whenever you are looking at the slide, two sections will be available, that is TS and LS. So whenever you will see the LS, at that time you can see the classical this one spindle shaped. But in case of TS, concentric appearance will be there. That you have to identify under microscope. Now, if you take the electron microscope, here T-tubules are absent. यहाँ पे T-tubules नहीं रहता है. So instead of T-tubules, what is present? Any idea? In case of smooth muscle, T-tubules are absent. ठीक है? But instead of T-tubules, what is present? Can you tell me? It is called as caveolae. Instead of T-tubules, in case of smooth muscle, what is present? Caveolae. Okay, so in case of smooth muscle, as I said, smooth muscle do not have T-tubules. Instead of that, caveolae are present. I will show you what is caveolae. Now, these filaments, they are same like the skeletal muscle. The striations are there. Okay, but if you see the arrangement of those actin myosin filaments, they are not properly arranged as they are present in case of cardiac muscle or skeletal muscle. That's why the striations are not present in case of smooth muscle. एक्टिन मायोसिन फिलामेंट्स रहता है, बट उसका जो अरेंजमेंट है वो पर्टिकुलर नहीं है, डेफिनिट अरेंजमेंट नहीं है, हैप्पैजार्ड अरेंजमेंट है. दैट्स व्हाई स्ट्राइएशंस आर नॉट सीन इन केस ऑफ स्मूथ मसल. Okay, here you can see, so this is the appearance. Here you can see the longitudinal section, okay, and this is the transverse section. Appearance अलग होता है, same नहीं होता है. So you have to identify which is the longitudinal section and transverse section. Transverse section में mostly you will see the concentric appearance. और इसमें कैसा appearance आएगा? Spindle shaped appearance आएगा. Now in schematic diagram it is very easy to identify, but under this 10x, okay, it will be very difficult. So you have to focus one or two particular cells and then you have to see the entire border of the cell to identify whether it is in transverse section or whether it is in LS section. Okay, so here you can see the caveolae. Instead of T-tubules, caveolae are present. Okay, and here for cardiac muscle and this one skeletal muscle, T-tubules are present. Next, this is the diagram which you have to draw in your practical notebook. ये diagram draw करना है practical notebook में, ठीक है? So here you can see two sections are there. Which section is this? Which section is this? Longitudinal section. Now, what do you think? What are the appearance of the fibers? Can you see the spindle shape? Yes. And this is transverse section, concentric appearance. ठीक है? तो ये mainly identify करना है. तो it is not that easy because यहाँ पे schematic diagram में separately दिया है. But when you will see under microscope, तो उसमें थोड़ा time लगता है. But still, it can be manageable. It can be identified. Okay. So that is all about the muscle tissue. Next, let's start with the trachea. Okay, so what do you know about the trachea? Now, okay, listen, before going to the trachea, there are basic things which you have to know about the respiratory system. So, respiratory system is having two parts, that is the upper respiratory tract and lower respiratory tract. I hope you have seen. Have you seen the trachea in the cadaver? Yes. Trachea? Okay, very good. Now, what is respiration? Everyone you know. Okay, then you know the two types of respirations are there: external respiration, internal respiration, right? Next, the respiratory tract is divided into two parts, that is the upper respiratory tract and lower respiratory tract. Okay, now what are the parts of upper respiratory tract and what are the parts of lower respiratory tract? Starting from the nasal cavity to the vocal cords which are present in the larynx, that is the upper respiratory tract. Vocal cord से नीचे जो part है up to the lung, these are the lower respiratory tract. Okay, so from larynx below the vocal cord, trachea, bronchi, bronchioles and lungs, these are the lower respiratory tract. Okay, and you can see the diagram also where the upper and lower respiratory tract are shown. So what do you think, trachea is a part of upper or lower respiratory tract. Okay, so in this term we will be discussing about the lower respiratory tract only. Upper respiratory tract will be discussed in head and neck section. When after this midterm, head and neck portion will be started along with the face. Okay, and along with the neck we'll be discussing about the nasal cavity, then about the pharynx, about the larynx. Not now. Okay. So now we'll start with the trachea. Now, do you know about this tracheobronchial tree? Anyone? क्या होता है tracheobronchial tree? How the trachea is divided and gradually how it is entering inside the lung. ठीक है? Why this is important? Because as you are going downwards towards the lung from trachea, structural changes are taking place. That means the structure at this level and the structure at this level is not same. Okay, so structure change hota hai, matlab histological changes are also there, starting from the lining epithelium, then cartilage layer, muscle cells, lymphatic tissue, everything, even the diameter also changes. Okay, so that's why we have to study the entire respiratory tract, mainly the lower respiratory tract, that also the histology. Why? Because as I said, the structural changes are taking place. That's why. Okay. So everyone, you know that trachea is divided into two principal bronchus. Trachea is divided into two principal bronchi, right and left principal bronchus, right? इसके बाद क्या होता है? Then the two principal bronchi will further divide into secondary or lobar bronchi. So 3 in case of right side, 2 in case of left side. Why? Huh? Why? You have not seen the lung. I know that you have seen the lung. Next, इसके बाद. Very simple. 3 means there are 3 lobes in the right lung. 2 means 2 lobes in the left lung. That's why the divisions are like this. Common sense. And the diagram is also there. See, I hope that day someone have shown you this one. Okay, तो क्या होता है जो प्रिंसिपल ब्रोंकाई होता है, राइट साइड में कितना डिवीजन होता है फॉर लोअर ब्रोंकाई? 3 डिवीजन. लेफ्ट साइड में 2 डिवीजन होता है. वाई? जो राइट लंग है उसमें 3 लोब होता है और जो लेफ्ट लंग है उसमें 2 लोब होता है. 2 lobes are there. That's why the divisions are like this. After that, what happens? You can see tertiary or segmental bronchi are there. Okay, tertiary or segmental bronchi. So that is 10 for both the side. 10 for the right side, 10 for the left side. Now they are supplying the bronchopulmonary segments. Till now we have not studied about bronchopulmonary segments, but you will be studying. ठीक है? You have studied? तो अच्छा है, तो पता है bronchopulmonary. How many bronchopulmonary segments are there? 10. 10 on right, 10 on left side. ठीक है? So for that you can see 10 on each side. Now this bronchopulmonary, this segmental bronchi will further divide into small bronchioles. So from here you can see bronchi to bronchioles. तो यहाँ पे एक jump हुआ है. So here also the structural changes are taking place. ठीक है? Next, these bronchioles will divide into terminal bronchiole, then respiratory bronchiole, then alveolar duct, after that alveolar sac, and then the alveoli. Okay, this is how the passage is there. Now, 2 parts are there, as you can see. This is the conducting part and this is the respiratory part. Okay, so this is the tracheobronchial tree. Next week, we will be discussing about the histology of lung. Directly, not the lung, will include this part also. Today up to trachea, and in the next week beyond the trachea, from starting from the 2 principal bronchi up to the alveoli. Okay, each and every part का जो microscopic structure है we'll be discussing along with the diagram, and this is quite difficult. Okay, this will be quite difficult. Now about the trachea, what do you know about the trachea? कहाँ से start होता है? Trachea is a tubular structure, right? Trachea is a tubular structure. So what is the length? Any idea? 10 to 15 centimeter approximately. From where to where it is extending? Okay, so it is the continuation of the larynx at the lower border of cricoid cartilage, that also sixth cervical vertebra. More or less it is present in the midline of the body. Okay, now after neck it is going where? It is going into the which part of the mediastinum? नेक के बाद thorax में आएगा, तो thorax में आएगा तो mediastinum से ही जाएगा, right? तो superior mediastinum. Okay, it is not going into the posterior mediastinum. Why? Before that only it bifurcates, it terminates, that's why. So can anyone tell me what is the partition between the superior and inferior mediastinum? Yes, at the sternal end. What is the vertebral level? T4. Okay, lower border of the T4 vertebra. So this particular trachea, it terminates at the lower border of T4 by bifurcating into two principal bronchi. Okay, that is the right and left principal bronchus. Okay, now this particular termination is for cadaver. In living individual it goes beyond T4 and it reaches up to T6 level in living individual, and in cadaver it is T4. Okay, next if you see the structure of the trachea, it is a fibroelastic tube supported by cartilaginous skeleton formed by C-shaped ring. Okay, so it is a fibroelastic wall supported by cartilaginous skeleton. Fibroelastic wall रहता है, and in between you will be getting the C-shaped hyaline cartilages. Okay, so throughout the length of the trachea 16 to 20 tracheal rings are present. Okay, and those tracheal rings are made up of cartilage. Which cartilage? Hyaline cartilage. ठीक है. Now, do you remember last time we have discussed about the hyaline cartilage? Now today we will use the hyaline cartilage. So whatever you have studied that day, today by looking at the slide, you have to identify the hyaline cartilage. Now posteriorly, so C-shaped रहता है, so like this C-shaped है, तो posteriorly gap रहता है. Okay, if it is C-shaped, posteriorly a gap is there. Now this gap is filled by a smooth muscle, and that is called as trachealis muscle. ऐसे सी शेपेड रिंग रहता है, सी शेपेड रिंग, तो जो पोस्टीरियर एस्पेक्ट है, दिस पोस्टीरियर एस्पेक्ट इज फिल्ड बाय ए मसल एंड दैट इज कॉल्ड ट्रेकियलिस मसल. और व्हिच टाइप ऑफ मसल इज दिस? स्मूथ मसल. Just now I have shown you the diagram of smooth muscle. So see again, you have to identify the smooth muscle fiber. Okay. Next, the trachea, the lumen of the trachea, the lining epithelium is called as respiratory epithelium. Okay, but what exactly is the respiratory epithelium? It is pseudo-stratified ciliated columnar epithelium. Okay, called as respiratory epithelium. Next, trachea is a tubular structure. Trachea is a tubular structure. So if you take a TS, what will be the appearance? Circular होगा. So see, this is the circular. So you have to identify which part is anterior, which part is posterior. How will you identify? Muscle कहाँ पे रहता है? Posteriorly. Okay, so see here the muscle is present. That means this is posterior side and this is anterior side. So anteriorly and laterally what is present? Cartilage is present, and posteriorly muscle is present. Next point, please keep that in mind whenever you are studying about any tubular structure. The TS of the tubular structure, उसमें layers रहता है. Layers will be there. Okay, here also you can see I have mentioned the layers. Now these layers, whenever you are writing it, you have to maintain the serial. That means either you should go from inside to outside or outside to inside. You can't mix. In the exam, if you are writing, if you are mixing it, if you are correct, then also you will not get marks. Please keep that in mind. तो कैसे लिखना है? Either inside to outside or outside to inside. Generally everywhere it is written from inside to outside only. Okay. So how many layers are there? 4 layers are there. So what is the first one? Mucosa. Then submucosa. Sub means below the mucosa. That's why it is called as submucosa. After that you are getting the cartilage and the muscle layer, and last is the adventitia. तो इसमें inner lining कौन सा? Innermost one? Mucosa. And outermost? Adventitia. ठीक है? That you have to identify under microscope. That is quite difficult. Okay, यहाँ पे तो लिखा है 1 2 3 4. स्लाइड में लिखा नहीं रहता है, इट इज नॉट रिटन इन द स्लाइड 1 2 3 4, that you have to identify. Okay, and that is the difficult part over here. So don't worry, I'll show you some images. Based on that, it will be very easy for you, okay, in first attempt to identify which is innermost side and which is the outermost side. Okay, now we'll go about the layers one by one. So first is the mucosa. Also, okay, so in the mucosa lining epithelium is present. From now onwards, please keep that in mind. कहीं पे भी mucosa आया, you have to mention about the lining epithelium first. Along with that, other cells will be there, but first what you will mention? Lining epithelium. Okay, now there is a shortcut for the trachea, that is respiratory epithelium. Respiratory tract है तो respiratory epithelium. If you are writing in the exam that trachea which epithelium, respiratory epithelium, you are correct. Okay, so trachea is lined by pseudostratified ciliated columnar epithelium. Now epithelium chapter you have studied, तो any epithelial tissue you know that basement membrane is there, तो यहाँ पे भी देखो resting in a basement membrane. Okay, provided with numerous goblet cells. Okay, so this appearance is very important which you have to identify in the lining epithelium. जो lining epithelium है respiratory epithelium रहता है, along with that goblet cells are there which secretes mucus. Okay, so how to differentiate between the goblet cells and the lining epithelium? गॉबलेट सेल्स जो होता है उसमें म्यूकस रहता है, ठीक है, तो ड्यूरिंग द प्रोसेसिंग वॉश्ड आउट, ये वॉश आउट हो जाएगा, सो इट विल नॉट टेक द स्टेन, दैट्स व्हाई इट विल अपीयर एम्प्टी, वाइट अपीयरेंस आएगा. So in the epithelial lining you will see white appearance or empty appearances are there. So those are nothing but the goblet cells. Next जो lining epithelium है उसमें क्या रहता है? Cilia रहता है. That can be seen under 10x also and under 40x. Not clearly, but you can differentiate that yes, cilia are present. Okay. Next, so as I said, cilia are present. Okay, so approximately 220 to 270 cilia are there. Now the mucus which is secreted by the goblet cell will entrap the foreign particle or foreign material which is going inside the respiratory tract. आफ्टर दैट जो सिलिया रहता है, सिलिया का जो बीटिंग होता है और जो मूवमेंट ऑफ द सिलिया, इट इज इन ए फॉर्म ऑफ ए वेव. टुगेदर ऑल द सिलिया आर बीटिंग एंड दैट इज इन द फॉर्म ऑफ ए वेव एंड दैट इज टुवर्ड्स द लेरिंग्स, दैट इज टुवर्ड्स द लेरिंग. तो क्या होगा, जो म्यूकस सिक्रेशन होता है दैट विल एंट्रैप द फॉरेन पार्टिकल एंड द बीटिंग ऑफ द सिलिया विल थ्रो इट टुवर्ड्स द लेरिंग्स एंड आफ्टर दैट विद द हेल्प ऑफ द कॉफ यू कैन स्पीट इट आउट. Okay, that's why the cilia are very important. Okay, and in case of chronic smokers, that beating is affected. So here you can see the mucus secreted by goblet cell entraps the dust and other foreign particles. Okay, now these entrapped particles are thrown away by the ciliary bead towards the upper part of the trachea and larynx, and they are ejected by cough reflex. That's why the cilia are present. Now please note that along with this lining epithelium and goblet cell, two more cells are there. Okay, first one is the brush cell. Note it down. Brush cells are there, BRUSH, brush cells and enteroendocrine cells. Okay, so these are the different types of cells present in the mucosa or mucosal layer. Okay, first you can see lining epithelium, then goblet cells are there, which already I have mentioned. Lining epithelium is the respiratory epithelium, pseudostratified ciliated columnar epithelium. Next, goblet cells. So what are the functions of goblet cells that I have mentioned? Next, brush cells are there, and enteroendocrine cells and basal cells. Basal cells are having stem cell function. कोई भी सेल अगर डैमेज होता है तो बेसल सेल कैन कन्वर्ट देमसेल्फ इनटू द अदर टाइप ऑफ सेल्स. ठीक है. नाउ व्हाट अबाउट द ब्रश सेल्स एंड द एंटेरोएंडोक्राइन सेल्स? ओके, जो ब्रश सेल्स होता है, ओके, दे आर हैविंग ब्लंट माइक्रोविलाई. They are having blunt microvilli on their surface, and they are chemosensory cells. So whenever any irritant chemical substance is entering into the respiratory tract, these particular cells can sense it, and they will produce the cough reflex against it, so that it can be thrown out. Okay, that is the function of the brush cells. Next, enteroendocrine cells, these are also a special type of cells, and they are secreting a chemical compound that is called as catecholamine. Okay, that is, now what is the function of the catecholamine? They are bronchodilators. During stressful inspiration, this bronchodilators helps. So when the bronchus will be dilated, more amount of air can pass through it. Okay, that's why these cells are also present. Okay, so this I think I have not mentioned, so whenever you are preparing the notes, please write it down. Okay, next, basement membrane. Which one? Don't worry, I will send the PPT. ठीक है, लिख लेना. Now basement membrane is there. So if you stain the basement membrane with the help of H&E, it present a glassy or homogeneous eosinophilic layer. That is the appearance of the basement membrane. Next, lamina propria is there. The lining epithelium, इसके नीचे basement membrane होगा. Just below that, a loose connective tissue is present. That is called as lamina propria. That is called as lamina propria. Now the speciality of the lamina propria is, in that lamina propria, bult is present. BALT. Now what is this bult? BALT, bronchus associated lymphoid tissue. Okay, as you can see, consists of loose connective tissue. Now what is this bult? It is nothing but the aggregation of lymphocytes and lymphatic follicles. Okay, that is present in the lamina propria, and this aggregation is called as bult, bronchus associated lymphoid tissue. Now along with that, in the lamina propria, these elastic fibers are also present. Okay, and these elastic fibers you will not be able to see under H&E stain. For that a special stain is required, that is called as oracin stain. Next, submucosa. Okay, तो submucosa में क्या रहता है? Mainly in the submucosa you will be getting the serous and mucous glands. Serous glands and mucous glands are present in the submucosa. That you have to identify in the slide. Okay, तो जो serous glands होता है उसका जो secretion है, that will keep the epithelium moist. और mucous gland का जो secretion होता है thick viscous, that will nothing but they will entrap the foreign material whatever is going inside the respiratory tract. And along with that, submucosa also contain some lymphatic follicles, blood vessels and lymphatics. These are also present in the submucosal layer. Lastly, the cartilage layer. Okay, so in cartilage layer, C-shaped rings of cartilages are there. Okay, that is made up of hyaline cartilage. So all the properties of the hyaline cartilage will be present over there. I don't know whether you have revised or not, but we have discussed in the class. Okay, and today I will see how much you have revised about the cartilage. Okay, next, on the posterior aspect, you will be getting the trachealis muscle. Now this is very important. In one field only, or in one field of view, you have to identify the cartilage, you have to identify the trachealis muscle. Okay, we have discussed both the appearance of the cartilage and the appearance of the smooth muscle. By looking at the slide, you have to identify. How will you identify? Which one is cartilage, which one is smooth muscle? How you will identify? हाँ बोलो बोलो. हाँ वेरी गुड. तो सी स्पिंडल शेप appearance will be there for the smooth muscle. और कार्टिलेज का कैसे appearance होगा? हाइलिन कार्टिलेज का. For that you have to study about the hyaline cartilage. Homogeneous appearance of the matrix. Then cell nest will be there. Lacuna will be there. Perichondrium is present. And lastly, the adventitia fibroelastic connective tissue, mainly blood vessels and larger blood vessels and nerves are there, and along with that some adipose tissue can also be seen. Now this is the diagram. This you have to draw in your practical notebook. Okay. Now we have discussed all the layers, right? We have discussed all the layers. Now try to identify which is the innermost, which is the outermost. कौन सा innermost है? Yes. So this is the innermost and this is the outermost layer. ठीक है. Now in the first layer, what you have to identify the ना yes mucosa. तो mucosa में क्या रहता है mainly lining epithelium. तो here you can see the lining epithelium. ये वाला lining epithelium है. This is the lining epithelium. Okay. That is the respiratory epithelium. ठीक है. In between the lining, can you see some empty spaces? Small. These are nothing but the goblet cells. ठीक है. Next here the magnification is less. That's why you can't see the individual cilia, but you can see the cilia. ये देखो. This is nothing but the cilia. ठीक है. Beyond that, can you see this small layer, loose connective tissue? This is called as lamina propria. This is the lamina propria. Beyond that, this particular layer is the submucosa. Submucosa में क्या रहता है? See, these are the glands. Submucosal glands are present. Submucosal tracheal glands, serous and mucous glands. Beyond that, which layer is this? ये कौन सा layer है? Cartilage layer. That's why I asked you, what is the appearance of the cartilage? Okay, this is the cartilage. ये देखो perichondrium दिखाई दे रहा है. ये देखो, you can see the perichondrium over here. Okay, so this is the cartilage layer. And here you can see smooth muscle, trachealis muscle. So please keep that in mind. The cartilage and the smooth muscle सेम लेयर में रहता है, ऐसा नहीं आगे पीछे. ओके, बिकॉज दिस इज द कार्टिलेज, यहाँ पे रहता है स्मूथ मसल. ओके, तो सेम लेयर में रहता है, नॉट आफ्टर इट और बिफोर इट. ठीक है, सेम लेयर. तो एज यू कैन सी दिस इज द कार्टिलेज एंड दिस इज द स्मूथ मसल. एंड व्हाट इज दिस? एडवेंटिशिया. ठीक है. So adventitia mainly larger blood vessel, nerves, along with some adipose tissue. All those things are present in the adventitial layer. Okay. So this diagram you have to draw in your practical notebook. ठीक है. Previous one, whatever I have shown you, that was the schematic diagram, and this is the actual diagram under image under microscope. तो इस पे identify करो. So which is the innermost layer? Yes. So this is the lining epithelium. Can you see some empty spaces? Yes. Goblet cell, and you can see the cilia also. After that, here small layer, this is the lamina propria. Then here you can see the submucosa. This is cartilage layer, and this is the adventitia. Now here trachealis muscle is not seen because in this particular view trachealis muscle is not present. ठीक है. So I hope it is clear. So first we'll take a gap of, it's 11:30. We'll take a gap of 10 minutes. After that, first we'll see the slides of trachea, and then we'll see the slides of muscle tissue. ठीक है. No one will go out. Sit and revise the trachea, whatever we have discussed earlier. आ जाओ, washroom में जाओ. তো এটা এখন 10x এ আছে. তো নেক্সট আমি 40x এ যোগ করব, তো লেমিনা প্রক্রিয়াটা ভালো করে দেখতে হবে. Between the cartilage and the smooth muscle, both can be seen. Difference between the cartilage and the smooth muscle. First you have to see all the layers, then specifically cartilage पे focus करो, इसके बाद smooth muscle पे focus करो. কার্টিলেজের মধ্যে অলমোস্ট টেনিংটা বেশি হয়ে গেছে একটা পার্ট আর স্টিল কার্টিলেজ কিন্তু অ্যাক্টিভ মাসলটা অ্যাক্টিভেট হচ্ছে তো হ্যাঁ স্পিন্ডল শেপ ওইটাই দেখার জন্য দেখি
left side is equal to that. Left side is equal to
अंदर जाओ
टेनेक्सी क्वेश्चन आता है ठीक है कोई एक फोटोग्राफ लेके रखो तो टेनेक्स में 48 सब ठीक है तब ठीक है आगे हमारे
अंदर आया बस फ्रेंड ने बाहर गया लेट के लिए खराब हो गया था ये खराब ही था ना
देख लिए जाओ जाओ
मटक ले तो क्या नाम देखो ना
देखो
खत्म, टाटा, बाय बाय।
भैया इसका वीडियो मत बनाइए, बंद कर दीजिए। नहीं बना रहे तो खराब कर दिया तो और कहीं नहीं डाल पाएंगे उसको। क्यों ही रिकॉर्ड कर रहे हैं सबका? लाइव नहीं चल रहा है।
देखना है ना।
नहीं लाइव नहीं, वो रिकॉर्ड करके वो यूट्यूब पे डालेंगे।
जब कोई अच्छा आदमी आता है उसका रिकॉर्ड किया हम आया किसका है
अरे सच में काट दिया।
और इसके बाद कार्टिलेज कार्टिलेज है इधर है इस साइड में
इधर से
हां हां
जो
कार्टिलेज
लाइनिंग
कार्टिलेज दिखाई दे रहा है, जो कार्टिलेज है जो स्पेनल से भी दिखाई दे रहा है। दो डायग्राम है कम तू जा लिखो इसको दिखा बहुत ज्यादा वैसे ये देख के निकलेगा तो दिखा देगा क्या मोटिवेशन वो डाल ले चैट
क्रेजी कर रहा है, क्रेजी कर रहा है। तो भाषा की किधर हम बहुत एकदम
फोर्टेक्स में है।